am Dr. Sunita Patra, Assistant Professor from Department of Botany, Government Nagarjun, Postgraduate of Science College, and my topic of this lecture is structure of protein. So, before starting our lecture, there is a disclaimer that the information I have taken from different known and unknown sites, and this is for study purpose. So, I duly acknowledge all the sources from where I have taken the information. So, we are going to start the structure of protein. So, before starting actually knowing the structure of protein, we should know what proteins are. Proteins are the most abundant organic molecules in the living organisms, and they contain about 50% of the cellular dry weight. They contain the fundamental basis of structure and function of life. Proteins are really very important, and they contribute towards a structural as well as a functional part of the life. So, we can see that in 1979 actually the word first it was described about protein. The scientists that described first about protein, what are proteins and where they are present. They are present in also in the protein. The word protein is derived from a Greek word proteos, meaning the first place. So, that means it is the macromolecule which we can keep in the first place because of its importance, because of its significance, because of its structural and functional significance in life. So, we are going to start towards today's lecture. What are proteins? The introductory part. The proteins, they are the nitrogenous molecules. They are composed of many amino acids. The amino acids, they are the monomeric unit. Now, before going to the structure of protein, first we should understand the classification, because proteins are of such diverse nature. So, first we have to study the proteins systematically, we have to classify the proteins. So, classification is based upon the composition and also based upon the hydrolysis products. After hydrolysis, what products are produced. So, based upon composition, we can divide proteins as fibrous proteins or globular proteins. So, fiber proteins, examples of fiber proteins are keratins, collagens, fibrils, etc. And globular examples of globular proteins are hemoglobin, insulin, etc. And based upon hydrolysis products, simple proteins we can divide it into simple proteins, conjugated proteins, derived proteins. Simple proteins, for an example, albumins and globulins. Then conjugated protein, we can further divide it into nucleoprotein, glycoprotein, chromoprotein. The example of nucleoprotein, we are also going to see in the future slides what are nucleoproteins. They are nucleins, then glycoproteins, myosin, and chromoproteins such as hemoglobin, chlorophyll, etc. Now, derived proteins, examples of derived proteins are proteases, peptones, peptides. That means after hydrolysis of protein, we get proteases, peptones and peptides, etc. This is also the classification of protein based on their chemical composition, simple, conjugated and derived proteins. So, we can also classify proteins as simple proteins, conjugated proteins and derived proteins, such as we have also seen in the previous slide. Simple proteins are globular proteins such as albumin, globulin, glutamine, prolamins, histones, protamines, etc. and also scleroproteins, collagen, elastins and keratins. Now, conjugated proteins are proteins in which one part is the protein and another part some other portion is conjugated with it. So, such as nucleoprotein, glycoproteins such as carbohydrate part, if it is attached with protein, glycoproteins, then mucoproteins, lipoproteins when lipid is attached with the protein molecules, phosphoproteins when a phosphate molecule is attached to the protein molecules, then chromoproteins and metalloproteins. Metalloproteins, there is a metallic part. Then coming to the derived portion, primary and secondary proteases. Primary is coagulated proteins, proteases, metaproteins. Then secondary part is proteases, peptones, polypeptides, peptides. So, this is the basic classification of protein, and now we can further move on. Now, again we are coming with the classification of proteins which is based on, again, composition is very important part. Then also one of the basic feature is shape of the protein. We are going to see each and every part, and functions. So, there are some basic features on which proteins has been classified. So, it is also classified on the basis of composition, shape and function. Now, based on composition such as simple protein, we have also seen in the previous slide also what are simple proteins that we have seen in couple of slides before, that there are simple proteins, then there are conjugated proteins, and there are derived proteins. So, what are simple proteins? A protein that contains amino acid only, that is simple protein. Simple, that is the constituent parts, the monomeric parts are only amino acids. For example, albumin, globulin and histone proteins, they contains only the amino acid parts, therefore, they come under simple proteins. Now, conjugated proteins. Conjugated proteins, what are conjugated proteins? Proteins having a non-protein part as a moiety. So, protein that have some non-protein moiety in its structure along with protein part. So, there are two parts, one is a non-protein part and another one is a protein part. So, we call such type of proteins as conjugated proteins. Then coming to derived proteins. Now, what are derived proteins? Derived proteins form simple and conjugated proteins by physical or chemical treatment. When we treat a protein with either physically or chemical treatment, we get some of the products. So, derived proteins are the derivatives or the derived parts or the products when a protein is treated physically or chemically, and the examples are peptones, peptides and denatured proteins. When we digest the protein with an enzyme or we heat it, because it is physical and chemical treatment also, and the products, they are known as the derived proteins. So, globular proteins, again the examples of globular proteins such as albumins, globulins, and also we have seen protamines, histone, we have already seen this in the previous slides also. The fibrous proteins are collagen, elastins and keratins based on shape. This classification, why I have shown these slides, because this classification is based on the shape, because we have taken one individual part. The classification of proteins, they have basic unit and we are taking one part at a time. So, this classification, this slide is the classification of protein which is based on the shape. So, some proteins, they are globular proteins according to their shape, and some proteins, they are fibrous in nature according to the shape. So, globular proteins, according to the shape, we can divide proteins into globular proteins and fibrous proteins. And examples of the shape, globular shape: albumin, globulin, gluten, protamines, histones, gluteins, protamines, these are, they all have globular structure, and collagen, elastin and keratins, they have fibrous structure. Now, this we can see further, we can I am elaborating the structure of protein. Fibrous protein, I have already said keratins. So, you can see the structure, the coil structure, and collagen triple helix, that means there are helical structure, 3 helical structure, they are intermingled and they are coiled with each other. So, these are the structure of fibrous protein. The diagram shows the structure of fibrous protein, and then the globular proteins, for example, myoglobin. Now, fibrous proteins or scleroproteins, again you can see the organization, how the helical parts, they are because we are dealing with the shape of protein, fibrous shape of protein. So, fibrous of shape of protein, you can see the diagrams as keratins and collagen fibers, how they are arranged to collagen, keratin and elastins. They are the examples of fibrous proteins. Fibrous proteins, they are involved. What are the functions of fibrous protein? Why they are so important? They have the structural portion. They have the structural significance. They are involved in structure, then tendons, ligaments, blood clots. So, all of these functions, they are very important. They are very significant. For example, collagens and keratins. So, each and every, you can very well acknowledge the function of tendons, the ligaments, the blood clots, which are very life threatening sometimes. So, contractile proteins, they are involved in movement, because without the contraction and this contractile, without the help of this contractile protein, you cannot even move your hands or legs. So, examples are muscles, microtubules. Now, where are these fibrous proteins present? So, they are present in cytoskeleton, mitotic spindle, cilia and flagella, cytoskeleton and then mitotic spindle. From where mitotic spindle, suppose what actually happens during cell division. Mitosis, when a cells divide, there are two phases, cytokinesis and karyokinesis. So, after karyokinesis, when the cells divide, when the nuclear part has been divided equally into two parts, and now it is the part of cytology or the cell to divide in two different planes. So, in that case, when the chromosomes or the chromatids, when the karyokinesis occurs, half of the chromatids they go to one pole and half of they are pushed towards another poles. Now, how these they are pushed toward the other poles? So, these are the spindle fibers. The spindle fibers they get attached with the centromere which are present in the chromatids, and these spindle fibers upon their contractile movement, expansion and contraction, they pull the chromatids towards different poles. And the same is the case with cilia and flagella. Cilia and flagella, they are the locomotory structures which are present in the lower animals and plant kingdoms, and these are the locomotory structures. Locomotory structures, they are present in antherozoids. They are in when we are talking about plant kingdom, antherozoids, because these are the male gametes which use the way which these male gametes, they in the aquatic condition they use water to move towards the female gamete part for fertilization. And also the animals, they also have cilia and flagella for their movement from one place to another. So, these are the locomotory functions, the structures. Now, structural proteins, another examples of structural proteins, because we are always dealing that proteins, they have significant role in structure as well as in the functional part. So, structural part is also very important. You can see proteins gives us shell shape, very significant part, strength, mechanical strength, as well as enable movement. Contractile proteins, they enable movement. So, we have also seen keratin, collagens and elastin, they are involved in these functions. And now, the example, structural example, hairs, everyone is having hair, then wools, nails, horns, hoofs, tendons and cartilages, they are all structural proteins. So, we can see each and every part, hair, wool, nail, horns, hoof, tendons, cartilage, everyone knows it very well what are the. So, these are the structural examples, structural proteins. Now, again I have also discussed fibrous protein, about fibrous protein. So, it is we can see that alpha keratin, the structure of hairs, the structure, then beta keratins, they have this spiral function, fibrous protein. Again, these are the examples of fibrous proteins. Again, now collagens. Collagens, they have repeating glycine and proline unit. Now, again hydroxyproline, what are the portion, what are the things which are involved in collagen? They are hydroxyproline, oxidized form of hydroxyproline, and it requires vitamin C, without which we can see scurvy diseases. These are the designs which I have taken from the PubMed. So, these are the structural animation part, how the collagen, the structural part, how these fibers they are intermingled with each other. Now, coming to globular protein. Now, most protein which move around, for an example, we always we daily come through it, albumin and casein in milk. Then these protein, what are the significance of the globular proteins? Globular proteins, proteins with binding sites. Significance is because this globular protein, they provide sites for binding. Site of binding for what? For enzymes. We can see that enzymes bind with their substrate. To substrate, for binding of substrate, there should be some binding sites. So, these globular proteins, when they act like an enzyme, when these globular proteins, they have functional significance, they provide binding sites. For hemoglobin, hemoglobin is a protein which stores iron and it transports iron. So, again it provides a binding site. Immunoglobulins, immunoglobulins, these are the proteins which are involved in defense mechanism. Immunoglobulins, so these are the quaternary structure of protein, immunoglobulin defense mechanism. Whenever we get through with the antigens, our body secretes certain types of protein which we call it as immunoglobulins. So, they also have binding sites. Then also very important protein which are present in our membrane, membrane receptor sites. Whenever there is a signal, there should be receptor molecule to receive this signal, and these signals they are further transmitted inside the cell, and accordingly the cell takes its decision. So, these globular protein have very significant role as they have binding sites for enzymes, they have binding sites for hemoglobin, the immunoglobulins and membrane receptor sites. Now, these are the this is the diagram of globular proteins. Globular proteins, you can see the folded nature, the spherical nature of these proteins. Examples are myoglobulin in a globular structures, you can see the compact structure. Now, this is structure of myoglobulin, again a globular proteins, it has hydrophobic effect. Then you can see the helix bundle there, then polar loops and also the non-polar core. Again the structure, there are different structure describe how this globular protein they appear. These are all animated portion. Again myoglobulin is a small monomeric protein and also it is involved in carrying and storage of iron. So, it has also a very significant role. Myoglobin, it is present in the muscle and the red color of this muscle is due to this, because it carries iron. It carries iron, it stores iron. This is small protein and it is a monomeric protein. Now, coming to conjugated proteins. So, we have seen conjugated, what is the meaning of conjugated proteins? Conjugated proteins are such proteins which have two parts, one part is of protein and another is non-protein part. So, non-protein parts for examples nucleoproteins, glycoproteins, mucoproteins, lipoproteins, phosphoproteins, chromoproteins and metalloproteins. So, we can see one by one. First one is the nucleoprotein. Now, nucleoprotein, what does it indicate? Nucleoproteins, example histone and telomeres. Where we get this nucleoprotein? We can see a eukaryotic cell, the diagram of eukaryotic cell, and eukaryotic cell has a very well developed membrane system. Now, what is the meaning of membrane system? That means each and every function is divided into simple compartments. So, every compartment had its own functions, such as mitochondria, it has its own function, chloroplast has its own function, and in the same way nucleus, which is the controlling part of the cell, it contains chromosomes. Now, these chromosomes, they are made up of histone proteins and DNA. The DNA, the functional part, which we call it as genes. So, we can see when we go to the nucleosome model of chromosome. Now, what we see there, the beaded structure. Now, these beads, they are the nucleosome, and nucleosome, they are consist of histone proteins. There are 5 different type of histone proteins which actually they are organized with the DNA to form the structure of protein. So, these histone proteins are of 5 types: histone 1, histone 2A, histone 2B, histone 3 and histone 4. Histone 2A, 2B, 3 and 4, each of them have 2 molecules and they form octamer, and these octamer, they are compactly arranged and we call it as nucleosome. And this DNA, they are 1 and 1/3 circle and they form the beaded string like structure and the compactness, and these are coiled and supercoiled to form chromosome structure. And there is another example, which is telomerase. Telomerase, the enzyme, these are the histone proteins. Histone proteins, they are involved in the structure formation of chromosomes. So, histone proteins have structural function. And telomerase, what is telomerase? Because when we go for chromosome structure, you can see there very well, the terminal part is known as telomers. Telomeres, that means what is the significance of telomers? Telomers, they have stability, they are meant for stability, and the telomere part of the DNA, it is replicated differently. That means when the whole part of the DNA is replicated, and telomere, the DNA which is present in the telomere, it is replicated by another enzyme, telomerase. So, we can see a telomerase enzyme there. So, nucleoprotein consists of histone proteins which have structure, it provides structure to the chromosome, and telomerase, which is responsible for the formation of telomers. Then glycoproteins. Glycoproteins are also very important proteins and very significant proteins. Why they are so significant? Glycoproteins of cell membrane. Now, you can see there are certain hormones, I have given example, follicle stimulating hormones and luteinizing hormones, but glycoproteins, you can see in the membrane there, we can see there are channels, membrane channels. These channels, what are these channels? These channels are basically meant for transport from ion from outside and inside, and through this ion channels there are the transportation of the membrane, it goes on. The first part, glycoproteins, which are the transmembrane protein. We can see transmembrane proteins means the proteins which you can see that through and through the membrane they are present in the plasma membrane, and receptor proteins. So, receptor proteins also you can see in the diagram. Then the receptor protein, it is very important that what type of signal is coming through, and the receptor they accept this signals, and they primary messengers, and they pass on this signal to secondary messenger. So, the membrane, and also for recognition of cells, suppose these glycoproteins which are the part of the cell membrane, the body it can recognize with the help of this glycoprotein whether certain cells, whether they are foreign in nature or they are own cells. Now, certain diseases it happens when own cells they are recognized as foreign. So, we call it as autoimmune diseases. So, recognition is also a very important part of this glycoprotein. So, glycoproteins, proteins in which a carbohydrate part is attached. So, such type of proteins they are known as glycoproteins, and they have very significant roles. Now, another is lipoproteins. Lipoproteins, that means proteins, one part is protein and another low lipid molecules they are attached. So, you have also heard about low density lipids, high density lipoproteins, low density lipoproteins. When we go for biochemical examination of the cell, you have to see what are the strategies, what are their level of LDL and HDL. So, accordingly the prescription is there, the medicines are there. So, we can see there the proportion of LDL and HDL. So, it also plays a significant role. We can see the proteins, the cholesterol, cholesterols, triglyceride, phospholipids are the part of membrane. So, triglycerides, cholesterol, cholesterol esters, proteins. So, lipoproteins also have the significant importance. Just one, I have skipped a portion of lipoprotein. Lipoprotein, when we go for cell membrane, the cell membrane is made up of lipid and protein. We say it is a unit membrane, lipid protein membrane. In the lipid, the protein molecules they are embedded, the fluid mosaic model of the protein. So, lipoprotein play a significant role in the structure also. Now, coming to phosphoproteins. Phosphoproteins, that means proteins which are phosphorylated temporarily for their regulation, a very important part. When any enzyme or a protein it is inactive in condition, they do not have a phosphate. As soon as a phosphate molecule is added to the enzyme, it becomes active, from inactive to active condition. So, we can see in the exam, suppose there is an enzyme A which is in inactive form, and as soon as a phosphate molecule is attached to that protein, it becomes active and moves on for its function. In the same way, we can also see enzyme B which is very active, inactive or active. So, it is vice versa, whether the enzyme, sometimes enzyme which is inactive and it becomes active by phosphorylation, sometimes enzyme B is active and when it is phosphorylated it becomes inactive. So, for activation or for deactivation of enzyme, it is very much important. Why phosphoproteins are, we can see example of cell cycle. Cell cycle means the cell it is divided into the phase of cell division and interphase. Interphase is a very long phase, and further it is divided into G1 and G2 phase, first gap phase, synthesis phase, G2 phase, and then phase of cell division. Now, each and when the cell moves from one part to another part, there are certain checkpoints whether it should move forward or not. So, whether it should move forward or not, who is going to tell? So, some of the proteins they are phosphorylated. There are proteins which are known as cyclin and cyclin dependent kinases, which became active while phosphorylation and cell moves in G1 phase. Further, there are cyclins and cyclin dependent kinases when the cell moves from G1 to S phase, and further from S to G2 phase, and the cone. So, there are certain checkpoints, and these checkpoints are highly regulated, and these highly regulated checkpoints they are regulated by phosphorylation and dephosphorylation of the protein. Now, these are the chromoproteins. Chromoproteins, that means proteins which have, suppose hemoglobin, you can see the red blood cells, then cytochromes, enzymes, you can see the enzymes containing heme particle, they are red in color, then you can also see chlorophyll, green in color. So, chromoproteins having certain type of color in them. So, color hemoglobin is red color due to it has iron. So, it gives red color to the RBCs. Then metalloproteins, metalloprotein again it has metal group. So, it is also very important for enzymes. Specific metals they are required for function of specific proteins. Now, these are the globular. We have we have talked about fibrillar proteins. These are hemoglobin and myoglobulin. They are examples of globular proteins. Now, both of what is the difference between the two diagram? Myoglobulin is a monomer containing only one polypeptide, and hemoglobulin we can see there are 4 subunits where there are 4 different polypeptide chains. They are attached together. Both of the proteins they are globular proteins. One is monomeric and another one is tetrameric proteins. Now, classification of proteins based on their functions. Protein functions. So, protein structure it determines protein functions. Now, what type of structure we should always know? We should always know that the structure always coincides with the function. Whatever the function of a protein is, the structure is accordingly. So, structure of protein determines the structure of function. Denaturation or inhibition which may change protein structure will change its function, because we can know that the structure of proteins, the structure of protein is accordingly formed according to its function. So, if ever there is denaturation, there will be inhibition in the function also. Coenzymes and cofactors in general may enhance protein structure. So, proteins classified by the function. Catalytic proteins they are the enzymes, then storage proteins they are albumin in eggs, casein in milk, zein in maize. Then transport proteins, hemoglobin it transfers iron from different part of the bodies, hemoglobin. Communication such as hormones, insulin and neurotransmitters they are the protein, and they are very important to communicate between different part of the cell, different each and every part of the cell it should be communicated very, because if we lose the communication we are the functional or we can see the diseases. And then contractile proteins such as actin, myosin, dyne in microtubules. Then protective proteins I have also discussed in the earlier slides. So, protective proteins are immunoglobulins, or we call it as antibodies. These antibodies they are secreted as our body gets an antigen, our body is infected with antigens. Then there are fibrinogens which are involved in clotting of blood. When we see if there is a cut and blood flows, there is formation of fibrinogen and it stops the blood from flowing. It is very important. Blood clotting factors. Then also there are certain very important type of proteins what we call as a toxin proteins, snake venom, deadly poisonous slave, neurotoxic, cytotoxic, these proteins, these are toxic proteins. And then structural. The cell membrane itself is made up of lipid and proteins. So, cell membrane proteins, keratins of cell, collagens, they are all involved in the structural part of the proteins. Contractile protein, you can see a man running, and these proteins they have significant role in actin and myosin. They are involved in this contractile protein. Whenever the body is in motion, you can see the contractile fibers in the muscle, how the muscles moves, contracts, and this the diagram shows that contraction and relaxation, whenever your body gets both of the part are important, contraction as well as relaxation. When it is totally relaxed, we call it is paralyzed. When it is contracted, we call it as tetany. So, contraction and relaxation, it is both of the part are very significant, and these are done by actin and myosin proteins which are present in the contracted fibers in the muscles. Transport proteins, again transport proteins, that means hemoglobin, they are the carrier of oxygen in the blood. Then serum albumin carries fatty acids in the blood. So, carrier transport proteins are hemoglobin as well as serum albumins, carrier of fatty acid in the blood. Then there are protein storage proteins, because when we are consuming some food, some of the part it is stored in such type of food. So, storage protein examples are different for different substances. Ferritin stores ferritin. Ferritin is the storage protein which stores iron in the spleen. Legumes and beans in the plants, they also have proteins, they have storage proteins, they store this protein as a food material. Then proteins and enzymes, you can see there are substrate and enzymes, many proteins, each and every biological step or the biological system, it is done with the help of protein and enzyme. Any of the metabolic pathway or any of the step, or we can say that it is not possible without the help of enzyme. So, many protein acts as biological catalyst or enzyme. Then thousands of different enzymes exist in body, catalase, amylase, trypsin, lipase, thousands and thousands of enzymes are there. Each of the enzyme, they have their specific substrate and they have their specific metabolic pathway. Then immunological proteins, we can see antibodies, gamma globulins. Whenever a pathogen enters the body, the immune system produces antibodies against it and it fights against that bacteria or that antigen to invade it. So, these proteins are very responsible, they are the defense mechanism of our body. Proteins, they also proteins as buffer, you can see proteins, plasma proteins and intracellular proteins. It is very important to maintain a pH of our blood buffer. So, therefore, protein acts as buffer, so that the pH should be maintained, because enzyme, they are very specific about their pH. The function of the enzyme is very specific about the pH, and if the pH of the body, of the cell, of the blood, it is disturbed, you can see the disturbance in the body. Proteins and hormones, they are transmitters. So, examples are insulin, glucagon, growth hormones, they are protein hormones. Then again we have also said protein channels and receptors. We have seen in the plasma membrane there are receptors, there are channels, and cytoskeleton. Cytoskeleton such as microfibrils, intermediate compounds, filaments, ligaments, microtubules. So, these are the cytoskeletal protein, they form the structure. So, proteins forming channels, receptors and cytoskeleton. Clotting factors we have discussed here. When we got a cut, a wound, clotting properties, it activates and make network or mesh of fibrin and it stops bleeding. So, what are the example, what are the clotting factors? Factor 1, 2, 3, 4, 5 etc. You can see the example how the fibrinogen, they make a mesh, a network, and ultimately the blood flow is stopped. So, these are the summary of what different types of proteins we have seen, the enzymes and their function. We have discussed it earlier. It is a summary of all the things. Now, vegan, what are the different source of protein? You also know that vegan, that means totally vegetarian food, starting from 42%, you can see from hemp seeds, then 40% in the pumpkin seeds, then 34% in the peanuts, then 28% in the almonds, 24% in cashew nut, poppy seeds, soya beans, tofu, and all fresh beans, spirulina products. So, highest number or higher percentage of protein is present in spirulina powder. 10 highest protein vegetation. So, 10 high vegetarian foods, protein per 100 grams we can see. It is paneer, soya bean, cheese, peanuts, legumes, date, nuts. So, you can see the percentage of proteins per 100 grams. These are the animal sources of protein. So, we have seen the vegan proteins, and again we can see animal proteins where such as shrimps, chickens, meat, egg, milk, fish. These are a very rich source of protein. So, thank you. So, in this part we have learned about what are proteins, classification of proteins, significance of protein and the sources of protein. So, acknowledges are due to all the unknown and known sources. Without which else the presentation would not have been so informative. Thank you everyone.